So without further ado, welcome to the Zoom the Scientist series. This series is sponsored by Lake Champlain Sea Grant, UVM Extension, and SUNY Plattsburgh's education program known as Watershed Alliance. Lake Champlain Sea Grant develops and shares science-based knowledge to benefit the environment and economies of the Lake Champlain Basin. Watershed Alliance aims to reach K-12 students and their teachers throughout the Lake Champlain Basin. Our goal is to increase awareness and knowledge of watershed issues in youth throughout Vermont and New York. The Zoom a Scientist series was created in response to the current need for more virtual programs. Every Tuesday and Friday from now until the end of June, we will be hosting a guest scientist to tell us more about their research in the basin and beyond. Just as a heads up, today's presentation is also being broadcast live to our YouTube channel and will be archived there for future use or to share this presentation with someone who couldn't make it today. Also, all of our previous and future sessions will be on the YouTube channel as well. So join me in welcoming today's presenter, Hannah Lachance, Knauss Marine Policy Fellow and the International Fisheries Science Specialist with NOAA. Hannah Lachance received her Bachelor's of Science in Animal Science and her Master's in Science in Natural Resources from the University of Vermont. She's worked in various areas of research and with various species spanning from genetics to ecology and moose to fish. Since completing her master's, Hannah has worked for Vermont Fish and Wildlife, USGS Lake Ontario Biological Station, and is currently a Knauss Marine Policy Fellow at NOAA, working as the International Fisheries Science Specialist. Hannah will be delivering a presentation today titled Cisco Science, Conservation and Restoration of Native Species in the Great Lakes. Native fish are critical to maintaining healthy ecosystems and healthy economies. In the Great Lakes, Ciscos, a group of native preyfish, have been identified as a top priority for conservation and restoration efforts. Hannah will talk about some of the research efforts designed to help inform conservation and restoration efforts for Ciscos in Lake Superior and Lake Ontario. The research focuses on the early life stages, eggs and larvae, and uses a range of field and laboratory methods. And with that, Hannah, I welcome you and I'll turn it over to you.
Thank you. Hi, let me just pull up my screen real quick. Share that. All right, well, thanks for having me. I look forward to telling everyone a little bit about some Cisco science. And so we're gonna dive right in, pun intended, because we've got a lot of really cool stuff to cover. And so let's start by talking about Ciscos. And Ciscos are sometimes referred to as a group of related species, some of which are pictured here. And that's because for a while they thought that they were actually all one species. And it's only recently that we've started to think of them as separate species. They are all native to the Great Lakes. And for those of you who aren't familiar with the Great Lakes, I'm going to try using my laser pointer here to point out the two that I'm going to be talking about today. So up here is Lake Superior, and down here is Lake Ontario. And Lake Ontario is bordered mainly by Canada and New York, and Lake Superior is bordered by Canada. This is the Upper Peninsula of Michigan, and it's also bordered by Wisconsin and a little bit of Minnesota as well. So those are the two Great Lakes that we'll focus on today. Also, I'm going to talk a lot about Cisco, which is this fish right here. So while the group of them are sometimes called Ciscos, which is plural, when I say Cisco singular, I'm generally talking about this fish. I'll also talk a little bit about some of its close relatives called kai and hoyi. You'll also hear me say bloater sometimes. So those are the fish that we're going to talk about today. And specifically, we're talking about them because, unfortunately, their populations have taken a dive over the past, well, I don't know, let's see, it's been a while. 60s or 70s is probably when they took their biggest hit. And generally, they used to be spread all throughout the Great Lakes in pretty good population sizes. And unfortunately, due to overharvesting, so humans collecting the fish for consumption and other purposes, And then also sometimes habitat degradation and other factors such as invasive species has caused their populations to go down in all of the lakes, even Lake Superior, where it looks like their population is still doing well, their numbers are nowhere near where they used to be. And so why do we actually care about this fish? That is an important question to ask. Cisco and their relatives are a prey species, which means that they are an important food source for other fish in that lake, or other organisms in that lake. And not only are they consumed as juveniles and adults, they're consumed at every life stage. And not only are they consumed at every life stage, but they're actually an important food source because their eggs, for example, are available during a time where other food isn't as readily available. Because Cisco lay their eggs right at the beginning of winter in about December, and they grow throughout the winter until they hatch in early spring. And so all throughout winter, they're a really key prey source for fish that don't typically have food available during that time. And then right as they're hatching out in spring, they're also some of the first food available for other fish. And then even as they grow, they're still a prey resource for many fish, including lake trout and salmon. And so that's why they're important to the lake ecosystem, but they're also important to the local and foreign economies. So because they're feeding fish that we like to eat, such as lake whitefish, which is popular in the Midwestern states that border the Great Lakes, and they're also important as a sport and commercial fish, which means that they bring people to the Great Lakes to go fishing for that fish, which means that they're spending money on hotels and renting boats. And so they're important to the local economy in that way. So because they're feeding these fish, they're indirectly important in that way. But Cisco themselves are also harvested because they're also very tasty. They can be, ice fishing is popular to collect Cisco, but also their eggs have become an important source of caviar in Northern Europe. So Northern Europeans have started to rely a little bit on the Great Lakes for providing them with that resource. So Cisco are not only important to the lake itself, but they're also important to humans and their economy. Which is why a lot of different agencies and groups have started to identify Cisco and their close relatives, such as bloater, as important for conservation and restoration efforts. So before we dive further into talking about Cisco, let's just talk a little bit broadly about what conservation and restoration is in general. So conservation is trying to prevent ongoing degradation. You're trying to save what's left. So the lakes that still have Cisco populations, we want to make sure that they stay there and that no future or current influences such as climate change or harvesting could continue to degrade those populations. Restoration is when you're trying to actively reverse such degradation. So let's say maybe their habitat is already destroyed. We're trying to actively improve that habitat. That would be more of a restoration. So we're going to take a pause before diving further into Cisco to kind of make sure that we are all understanding what conservation and restoration is. So I'm going to show you two videos, and the next question will be guessing what the project in each video is geared towards. Is it geared towards restoration? Is it geared towards conservation? Or maybe it's geared towards both. So the first video, I'm going to click and hopefully you all can see it. I'm going to skip ahead a little bit. This is a video showing the stocking of bloater, which is a cousin to Cisco. And there, these little silvery fish you can see are getting blown out and they're getting stocked. So those fish were reared in a hatchery setting and fed a pretty good diet so that they would grow fast and have the best chance of survival once they were put into the lake. So I'm going to pause it there. So that's the first video. The second video, here we go. So this is a video of that we took in Lake Ontario. Both videos are from Lake Ontario, where we were taking a look at the bottom habitat of the lake. And so we are trying to see what kind of material is down there, how clear the water is, and see basically what quality habitat is available. And in this video, I just want to point out, we'll watch it quickly one more time. As we go down, you can see a couple of rocks and a lot of these white fragments. And those are actually shells, which have, which are a lot of what you see on the bottom of Lake Ontario nowadays. I'll also note that when the frame hit the bottom, you notice that it hit hard material. So that gives us a clue that it's hard substrate, not mucky, silty material on the bottom. All right, so we are actually gonna be moving on to our first poll question. So I want you to vote on what you think each video was an example of. Do you think video 1, where they were stocking the fish, is an example of restoration? Video 2 of conservation, or vice versa? Or C, is video 1 restoration and video 2 both conservation and restoration? Or D, as video 1 conservation and restoration and video 2 conservation? Just to remind you, video 1 is where they were stocking fish, and video 2 is where they were assessing habitat. I'll give you a second to vote, and then I will reveal the answer. Okay, so just for the sake of time, I'm going to reveal the answer, but keep voting if you are in the process of doing so. I would say that C is the most correct answer. Video 1, where you're actively adding fish back to the lake, that's definitely geared more towards restoration. Video 2, it could go either way. We could use that habitat information we're collecting to try to figure out if there's good habitat available, and if so, how to conserve it. Or we can notice that the habitat maybe isn't so good and start planning restoration efforts. So that was a little bit of a trick question, but it was intended to show you that a lot of times conservation and restoration efforts go hand in hand. We're trying to save what's left and restore what has been damaged at the same time. All right, so moving on. The next question is, how do we actually decide what we need to do in order to conserve and restore a species? You know, we just showed 2 examples, one where you're stocking fish, another one where you're looking at the habitat. How do we decide even that those projects were necessary? Well, you generally bring experts together and have them discuss what we know about that species and also what we need to know in order to conserve and restore that species. And these are just 2 reports that were generated from such meetings. Ellen George is listening in. I'll give you a quick shout out because she helped organize this one over on the right, where that was geared more towards identifying research priorities in Lake Ontario specifically. And that's because it's great to know as much as we can about the species across the Great Lakes, but also Cisco and Lake Ontario might need different things than Cisco and Lake Superior. So having these conversations for each lake is important. But just an example of what has been identified in some of these meetings is we've identified that we need to identify, I'm saying that a lot, any impediments to reestablishment and growth. So that basically means what could possibly lead to the success or failure of this species. For example, is climate change gonna positively or negatively impact this species? We could also identify gaps in knowledge of life history and genetics. So we know a decent amount about these species, but there's still a lot that we don't know. And one of the biggest hurdles to get over is trying to identify the different species. So at the beginning, I showed you a picture of a lot of very similar fish. And as I said, we used to think that genetically they were very similar, and visually, sometimes, especially in the early life stages, they look very similar. But they hold different roles in the ecosystem. Some live deeper in the lake and eat different things and are eaten by different things. So they're filling a different part of the lake ecosystem than fish that are in, let's say, closer to shore or further up in the water column. So identifying the different species through genetics, perhaps, and then being able to learn more about each species' life history is important so that we can learn how to conserve and restore each one catered towards their specific needs. And then another thing that has been identified through these meetings is understanding the role of genetics and environment on the development of a phenotype. And that's basically saying that, well, even within a species, let's say just the Cisco species, they might have similar genetics, but if a Cisco is in an area of the lake where it's warmer, for example, maybe their genetics are being used slightly differently than a fish that's in a colder part of the lake, for example, because the environment helps tell you when to use certain parts of your genetics. So understanding the differences in that perspective is also important. Which brings me to some of my research. So I am just one of many folks working on this project, and the areas that I was able to contribute to was through some of my graduate work, looking at climate change impacts on Cisco early life stages, and then also looking at the genetics and life history of Cisco in Lake Superior. And then after graduating, I worked for USGS before shifting to DC for my current fellowship. And while there, I worked in Lake Ontario and helped to try to identify which habitat Cisco like to use when laying their eggs. So without further ado, we're gonna continue to swim along and we're gonna start by talking about climate change. So climate change is a really big question to tackle. And so we need to break it down or frame it around Cisco and particularly the life stage and location that we are asking this question for. So we're gonna zoom into the Great Lakes, and then we're actually gonna zoom into winter particularly, because that's when they're laying their eggs and larvae. And we'll talk a little bit more about this in a second, but the eggs and larvae are the early life stages. Larvae is when they first hatch out from the eggs. And knowing what happens during those two life stages is really important because making it to adulthood is really hard for Cisco or for fish in general. And once they make it to adulthood, they have a better chance of surviving. But getting past those early life stages is really where we want them to succeed, because then they will have individuals entering the population and helping them to, go on and make babies themselves. So we're focusing on winter in the Great Lakes because that's when the eggs are incubating. And specifically, if we think back to their life history, they lay their eggs at the beginning of winter, generally when ice is forming on the lakes, and they hatch generally at the beginning of spring when the ice is melting off the lakes. So we're not only thinking about winter, but we're starting to think specifically about how climate change could be impacting ice in the Great Lakes. And another reason why we're thinking about this is, like I said, if Cisco or any fish generally can make it to age 1, which is when they're starting to be adults, they have a better chance of survival. And so here on the x-axis, let me get my little pointer again. We're looking at maximum annual ice coverage. So over here is where they had, at some point during winter, 100% ice coverage. And here they only had about 5% ice coverage. And then on the y-axis, this is the density or amount of Cisco that make it to adulthood, age 1. And so you can see that many years, they don't have a lot of success with the fish making it to adulthood. But there are a couple of years where they had really good success. And most of those years happened to occur when there was a good amount of ice on the lake. Obviously, that's not the complete recipe to a good success of fish making it to adulthood, because there are plenty of years where there was good ice and the fish didn't make it to adulthood. But ice does seem to be an important factor, which brings us back to thinking about how climate change could be impacting ice in the Great Lakes. And this is a really cool GIF by NOAA, where it shows the maximum ice coverage each year since the 70s up until the present. And while you can see that each year it varies, overall, there's unfortunately a significant decline in ice coverage, and that's predicted to continue. So we've taken our question and started to filter it down, but we can filter it down just a little bit more. So thinking about ice, if there's ice present or if there isn't ice present, it changes other environmental factors in the lake. So it can change the temperature, it can change the amount of light that's reaching through to the eggs, and it can change the flow. And other researchers have been looking into temperature and flow, so that kind of left light for us to be pondering. And if we think about it, when there's no ice and in the shallower areas where Cisco tend to spawn, we see that 100% of the light can reach the areas where Cisco are. But if there's ice coverage, only about 35% of the light can reach them. And if there's snow on top of that ice, only about 7% of the light can reach the eggs. So Cisco are going from developing their eggs in an environment that's typically pretty dark when there's ice to nowadays being more and more exposed to light. And how is that going to affect either their development rate, how quickly they grow, or their overall survival? And so that brought us to our specific question. We started with how does climate change impact Cisco, and now we filtered it to how does light impact Cisco, eggs, and larvae. So we're gonna continue along, and how do you do that? Well, first we gotta get some Cisco eggs. So one of the graduate students working on this project with me is Taylor Stewart, and he got to go to Lake Ontario and help with our partners, New York DEC and Tunisian Laboratory of Aquatic Science, which is a USGS facility. And he got to help collect some males and females right when they were about to lay their eggs. And when they're ready to lay their eggs, you can actually just run your hand along their belly and squeeze out the eggs into a bowl. And then you can also do the same with the male and put their material in the bowl, mix it up, fertilize some eggs. And then we took those fertilized eggs and shipped them back to Vermont, where we reared them in one of 3 treatments. Just to kind of preface this, not a lot was known about how light affects fish growth. So we started with extreme treatments, and this is the pilot study, which means it's like a first attempt of an experiment where we just start with some basic treatments. We only had 1 tank. Usually you want to have a couple to limit any confounding variables, which means if something went wrong with 1 tank, it didn't mean it was an effect of the light, it was just a tank thing. But we won't get into that. But basically this was a really good first pass, but it just mostly informs future research. So our 3 treatments were light, dark, and a regular day-night cycle. And everything else was kept similar. The flow, the temperature of the water, everything else was kept the same. So we were really just testing the effect of light on these eggs. So as I said, Taylor Stewart was a great help in this project, and he helped monitor their development and other factors by taking pictures of them on the microscope as eggs, and then also after they hatched as larvae. So not only was he keeping track of development rate, he also was measuring yolk sac volume. So if you think of like a chicken egg, that yolk is actually a really important energy source for the chick as it develops. And in the fish, it's this area, it's a little hard to see, but it's something that they feed off of as they're developing as an egg. And then as a larvae, they actually still have a little bit left, and they're feeding off of it until they learn how to feed off of particles in their environment. So understanding how much of that yolk sac is available is important. And then also monitoring mortality. As I said, this is a rough time of life for Cisco. There's a lot of natural death that occurs, but any additional death that's occurring due to exposure to light or dark, we wanted to be able to monitor. And so in addition to the traditional methods that Taylor brought to the table, I kind of brought some of my past experience with genetics to help complement and help us get a deeper understanding of what's going on. So it can get very complicated in the molecular world, but basically what's happening is there's a couple different levels you can look at. In the DNA, if you look at all of the DNA, you're able to look at all of the genes that you have available to you to make anything that you need throughout your entire life. But you're not using all of those genes at the same time. So we really wanted to know which genes the fish were using and at what level. And in order to do that, we looked at the RNA, because you have a gene, and if you want to use that gene, you make a copy of it with the RNA, and then that RNA goes to create a protein, which builds up what you need for a functioning organism. So by capturing the RNA, we're looking at only the genes that are being used at that time and how much of that gene is being used. So here, for example, we can tell that genes 1, 4, and 7 are being used because RNA is present. But if we gather all of the RNA, then we can tell that gene 7 is being used a lot. So knowing that that is in high use is important because maybe in, let's say, the light treatment, we're using that gene a lot. And in the dark treatment, gene 7 still might be used, but at a lower level. Let's say you're only getting a little bit of the RNA. So knowing that that gene is being used and at what level is important to understand how the environment is influencing which genes we're using. So to do this part of the project, we focused on eggs and larvae right as they were hatching from the light and dark treatments. And for the results, first of all, we noticed that the fish reared in the light treatment hatched a full week before the other 2 treatments. So they hatched quicker. We also saw that they had a way smaller yolk sac, so less of that energy reserve for the fish reared in the light treatment compared to the other 2 treatments. And then we also saw mortality was fairly similar for the eggs, but much higher for the fish that hatched out of that light environment. Again, this is a pilot study, so again, this is just a first look at what possible implications light could have on these fish. But knowing the beginning of the story through the traditional methods is really important. And now we can start to think about the transcriptomics or those molecular methods that I mentioned. So the main takeaway from this slide is that we did see different genes being expressed at different levels compared to the light and the dark treatment. So upregulated means that these genes were being used at a higher level in the light treatment than the dark treatment. And downregulated means they were being used at a lower level in the light treatment than the dark treatment. So that's interesting, but what we're really curious about is what do those genes actually do? And obviously there's a large number of genes right here, so we won't go through each of them, but I'll give you an example. So one of the genes, BVRA, was used, being expressed in both the dark and the light treatments. And so over here, the number of reads, that's basically the number of copies of RNA. So the gene was being used in the dark treatment, but there were way more copies of RNA in the light treatment, which means they were using more of that gene in the light treatment. So what does this gene actually do? Well, genes can have more than one function. And one of the functions of this gene is helping to break down red blood cells. But more interestingly, it's also part of the insulin signaling pathway. And for any of you who have family members or yourself or friends that have diabetes, you might know that insulin is actually important in uptaking or using glucose and fatty acid. And if we think back, that yolk sac that I mentioned is actually mostly fat. So that starts connecting the dots between what we're seeing in the molecular methods and the traditional methods. So here we're seeing the increased expression. So they're using more metabolism pathways. So they're using more genes that help break down energy or burn energy. We're also seeing that their energy reserves are depleted. They're smaller yolk sac. And then unfortunately, we're seeing a higher level of mortality. So again, it's the first level of an experiment. There could be more work into this, but it's starting to connect the dots on possible implications of light on these early life history stages, which can get our brains starting to think about what that means for conservation and restoration. So that's the first project. Moving on to the second project, we started to think mostly about that second life stage, that larval life stage from right when they hatch out of the egg until they disappear into the next life stage called the juvenile stage. And we know a little bit about that life stage by rearing them in captivity or capturing them maybe once a year in the lake, but we really wanted to know how they're doing in the lake from right when they hatch until they disappear in the next life stage. And so we went to Lake Superior for this project, and this is a picture taken during some of our field work in the Apostle Islands, which is a beautiful area. So again, I just want to point out Lake Superior is this big one up here. And also I want to point out that while for the first project, we were talking just about this fish, Cisco, we're now going to start talking about some of its closer relatives, like Kaiai and Hoiai, which are smaller fish that have these deeper bodies, and they tend to live deeper in the lake. I'm also bringing this slide back up to point out that these are pictures of fish as adults. And visually, even if you're not used to looking at fish, you can probably tell them apart because you can look at their mouths and see that some of them have different shapes. Their sizes are different. Their body shape is different. So there are visual ways for you to tell the adults apart. However, when you get to this larval life stage, it's really hard to tell them apart. And some folks try to tell them apart visually and make mistakes because it's just really hard to do. So when we're trying to learn more about this life stage, the first thing we need to be able to do is to tell them apart. Because if we scoop up some larvae and we start to measure how long they are, figure out when they hatch, calculate how quickly they're growing, and we start to think we're painting a nice picture for this species, but it turns out to be 3 different species, then we don't really know as much as we thought we did about this species. And it could lead us to wrong decisions to make when trying to conserve and restore this species. So the first step for this project was to try to find a way, using genetics again, to tell these species apart if there are different species present. And to do this, we were in Lake Superior, but focusing on one region called the Apostle Islands, because we wanted to sample that area every single week across that entire life stage. Once we are able to identify them, then we can start answering some of those life history questions, such as when are they hatching? Where are they hatching? What are their lengths at each week, and therefore we can possibly calculate how quickly they're growing, et cetera, et cetera. So with the help of a wonderful technician, Verena, we got to go to Lake Superior and sample every single week using this type of a net, which is called a bongo net. And you pull it right along the surface of the water because that's where the larvae like to hang out. That's where their food is. That's where the temperature is right for them. And every single week we went to, with the help of Verena, 10 different sites and sampled from the time that the ice melted out and we could get the boat out, which was mid-May, it was a late ice off season that year, until mid-July when we stopped catching larvae in our nets. And I just want to bring up the idea of scale here because the Great Lakes are huge. And so while this looks pretty close to each other, these are all actually fairly distant points from each other. So we were trying to figure out if larvae were hatching at different times off of different locations, if there were different species in different locations. So that's part of what we were looking at with these 10 different sites. So once we got the larvae, we took their picture with a microscope so that we can measure along and see if week 1, the lengths are different than week 2, 3, and 4, etc. And then Verena did a project where she was looking at what they were eating over time. So she did a very delicate surgery because these baby fish are just a couple millimeters long. They're really small. So she developed some surgical skills and removed their stomachs and looked at their what they were eating. And then the rest of the individual went to me so I could figure out the genetics and tell them apart. So just to give you a little bit more background, there is, at this point in time, before we looked into more genetics, there was a way to tell whitefish from non-whitefish. And whitefish is one of those close relatives that we've been showing in those pictures. And so while it's great to be able to tell whitefish from the other relatives, we still can't tell the other relatives apart from each other. And how this method works is we take basically one gene and we use an enzyme that cuts that gene based on the sequence. So if the sequence, or basically the series of A's, T's, C's, and G's, is different, which it is from whitefish to others, it'll cut it in different spots. And then it'll cut, a different number of pieces and different lengths of pieces. And that's how you can tell them apart. So that's great. But like I said, we want to tell the rest of the relatives apart. And so maybe we need to look at more than just one or a couple of genes. We need to look at as much of the genome as we can. And the genome is all of the DNA. So before we dive into that, we're going to play a little game of who's who. So this is our next poll question. And as I said, it is very hard to visually tell these apart, but I just figured it'd be fun to try to tell, see who could guess which one's a whitefish. And unfortunately, before I sent this question in, I realized I made a mistake. Anytime you see Cisco, technically, it's a non-whitefish, because at this point, that's all we can tell, whether it's whitefish or non-whitefish. So for A, if you think the whitefish is the one on the left, Go ahead and vote that. If you think the non-white fish is on the right, if you think the one on the left is not a white fish and the one on the right is, vote B. If you think both of them are not white fish, but still in that relative group, vote C. If you think both of them are white fish, vote D. If you think neither of them are white fish or non-white fish, maybe they're a completely different fish, go ahead and vote E. So I'll give you a second to do that. And while I do that, I'll actually go to the bigger picture here just so you can get a better look at them. And just for the sake of time, I'm going to keep moving along. There we go. So it looks like most of you thought that the white fish was on the right. I think my computer's a little delayed. And it was actually the white fish is the one on the left. And I was playing this because I thought it'd be a fun little game, but also because we did try to visually ID some of these fish, but we also checked it with genetics. And in doing so, this fish that was identified as a Cisco initially was actually genetically identified as a white fish. So that just kind of goes to show that it's really hard at this life stage, and using genetics is the way to go to be able to tell them apart. So that brings us back to that bigger genetic method where we looked at most of the genome to be able to try to understand any possible differences to tell the species apart. So we're not going to vote on this one, but it's basically another game of who's who, except this time we have more tools and we can actually tell them apart. And so what previously was thought to be 1 or 2 species in this area was actually 3 or 4 species. We found a couple of whitefish, and then by focusing on the non-white fish, we found Cisco, hoyi, which is also called bloater, which you might recall from earlier, kai, and then we found a couple of hybrids, which means that these fish were mixing and having some babies as well. So this slide talks a little bit more about how we can tell the species apart, but again, for the sake of time, I'm going to actually keep moving along. But basically, we compared the genetics to adults where we can visually tell which species it is, and it definitely told us that we were having 3 different species. Now we can start answering some of those life history questions, such as when are they hatching? And we saw that Cisco or Artidae was hatching very early in the season. And then the next week you start to see a couple of the kai pop in. And as time goes on, you see less and less Cisco because they're growing up and starting to move into the next life stage. And right as Cisco are starting to disappear is when you're starting to see more of that 3rd species, hoyi or bloater, appear. And again, as more hoyi appear, less kai are there because they're growing up and starting to move into the next life stage. So knowing that they're hatching and growing at different times is really important. This basically shows the same thing, except it's showing length. So basically, it just kind of shows that they're small, then they grow bigger until they disappear, but also that they're appearing at different sizes. Artidae, for example, is showing up smaller than kai is. So kai is hatching a little bit bigger than Artidae. And that's just something else that we were able to learn. And then finally, that yolk sac that they feed off of, we found a lot of little Artidae with that complete yolk sac, but we found the 2 deeper water species, hoyi and kai, had less of that yolk sac when we were catching them at the smaller sizes. So either they were burning through it earlier, or maybe we're not catching the smallest size available. Maybe there's still an earlier hatchling that we haven't caught yet. So that's just identifying another area that we should look into. So the biggest conclusion from this project is we can finally tell these little baby fish apart, which allowed us to look into a lot more and learn about that early life stage, which will help us to understand how to help that early life stage. So again, for the sake of time, I'm just going to keep moving along, but I'll happily answer questions at the end. So this is the 3rd and final project. This is one that I worked on just this past winter with USGS in Lake Ontario, which is where I worked for a little bit before moving to DC. And so here we're looking for what habitat Cisco like specifically for laying their eggs. In Lake Ontario, they used to spawn or lay their eggs in a lot of different locations. Nowadays in the US waters, we're pretty much finding them in this one region, and specifically in this one bay is where we're finding most eggs. In the Canadian waters, they're mostly finding them over here in the Bay of Quinte, but for today, we're going to focus on this area called Chameau Bay, because that's where we have known that Cisco are laying their eggs. However, recently there was a survey done where they scanned the whole lake looking for larvae, those baby fish. And it's a little hard to see, but everywhere that there's a blue dot is where they found those baby fish. So for example, here's one, and here's one, here, 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 here. So basically we're finding larvae in a lot of different locations, which kind of made us start to think maybe Cisco are laying their eggs in locations that we did not expect. So we need to start expanding what we thought we knew. Like for example, whoops, here we go. There we go. So this is just that one bay in the US, and here's where we were finding most of the eggs. And this is a shallow part of the bay. So it comes up to about sometimes even like 4 meters, which is pretty shallow. And it's generally rocky as well. And previous research has shown that that's what Cisco tend to like, is shallow and rocky. But if we're finding larvae in all parts of the bay, then maybe we need to be thinking beyond just shallow and rocky and seeing if they like maybe deeper or different kinds of substrate. So again, what we thought we knew might not be true. Maybe they're going in other locations. And what's driving that? Maybe they prefer this, but they also are okay with this. So maybe they'll put most of their eggs over here and a couple over here as well. So that caused us to want to go out and investigate more. But of course, this is the time of year that Cisco like to spawn. They like to come in right as the ice is forming, which makes it difficult. And while we would have loved to look at all of Lake Ontario, we decided to focus within that one bay where we know they lay eggs, but look in multiple places within the bay. So everywhere here that's red is where we knew that there were eggs, and everywhere that's blue is where we set traps to look for eggs. And the different lines within the bay are just showing different changes in depth. So here is shallow and rocky, and we set a couple traps in the locations where we know we get eggs, and then we set some in deeper areas close by. We also set some in near shore and offshore areas across a range of different substrates, which is basically whether it's rock or mussel shells or weeds or mud. So we tried to get a whole range of habitat types to see where we would get eggs. And this is just a quick example of the different kinds of traps that we use to collect eggs. The eggs could drift into this trap or onto this mat. And what we did was we clipped these different traps onto a long piece of rope or line, and that would allow us to sample multiple locations in a gradient of depths and substrate types. So this is just preliminary results because we're still processing data that we collected. And basically we found eggs almost everywhere that we set traps. Now we found different numbers of eggs, and so to better show you that, I'm going to zoom in to this location. And this location includes this shoal right here, which is where we knew that Cisco were laying their eggs. And this color gradient is showing that green and yellow and orange generally means high numbers of eggs, but purple means low numbers. And where there's an X means we didn't get any eggs. So if you look in this shallow area that we knew they were laying eggs, we're seeing a lot of eggs. So they're not only laying eggs there, they're placing a lot of eggs there. In this deeper area just close by, we're not seeing a lot of eggs, if any eggs, but they are laying eggs there. And then here, this is another shallow area. We're also seeing a decent number of eggs, but again, close by where it's deeper, we're not seeing as many eggs, but we're still seeing some eggs. So I'm going to skip past this one. This basically is showing some of the preliminary data. So the x-axis is depth, so shallow to deep. And the y-axis is egg density, so how many eggs. And so you can see that we're seeing the most amount of eggs in the shallower areas. So generally from about 1 meter to about 6 meters, we're seeing a lot of eggs. But we are also seeing some eggs in the deeper waters as well. The color here is showing the different types of substrate. And anything that's gray, we don't know yet because we're still processing the video footage we took at each location. But you can see that there's a range of substrates that they're placing their eggs on. And it's not, at this point in time, really showing a strong preference. So they might be more preferential towards the depth and less caring about the substrate. But this is just a preliminary guess. We need to continue processing the results to really know. So this brings us back to thinking about conservation and restoration as a whole. We have now been able to help identify any possible impediments to reestablishment and growth by looking at climate change, habitat preference, trying to understand what they want and what could be leading to success. We also filled in some gaps in knowledge, such as we can tell those fish apart now, which means we can tell more about that life stage or that life history at the larval stage. And then also understanding roles of genetics and environment, that goes back to that climate change question again. Environmental factors such as light do change how the fish are using their genetics, and that's important to know. So there was a lot of people that helped with all of these projects. I don't have time to list everyone individually, but I want you to know that this is a collaborative effort, which means a lot of different partners and scientists and stakeholders came together to make this research happen. And I couldn't have done it without anyone. And these are just my projects. There's a lot of great projects going on in the Great Lakes, trying to help understand and inform the conservation and restoration efforts for these important group of prey fish. So with that, we'll move on to questions. Thank you all.
Awesome. Thanks so much, Hannah. I learned a lot. So I appreciate you putting this together and setting aside the time to join us. So folks, if you have questions for Hannah, go ahead and open up the Q&A box and place your questions in there. And please stick around if you have the time for a final feedback poll and a take-home activity. So Hannah, I'd just like to start off and ask you a question that we've been asking all of our presenters, which is if you can kind of put yourself back into maybe the middle school, high school aged mind of young Hannah and think about sort of what first led you down this path of wanting to get involved in natural resources and conservation and restoration and just sort of studying maybe fisheries. So if you can kind of maybe walk us through what led you to this field of study in this professional field.
Sure. So that's an interesting question. Some folks know what they want to do since they were young. I was not one of those people. I have a large range of interests. And so as, let's say, a middle schooler, I think probably my career choice, if anyone asked, would be a teacher. Or I think I briefly wanted to be an author, but that didn't last very long. I just changed my mind on that. But I have always enjoyed being outside, whether it's just playing in my backyard or going on walks. I've always loved being by water, but I would say I didn't have a strong interest in fish until more recently. So if you don't know what you want to do at this point in your life, that's okay. But a lot of your hobbies, you will become an adult and reflect back on and realize that they did kind of unintentionally bring you to perhaps where you want to be career-wise. So for me, it was more identifying that I really liked working with animals, and then I realized I liked research, and then I realized that I particularly liked research around wildlife and then conservation and restoration and fisheries. So it kind of became developed and honed in over time for me.
Awesome. That's great. Thank you for that. I think it's always interesting to hear folks reflect on sort of the, their momentum into their given field. So it's always interesting. I had no idea what I was going to end up doing either. So I was, we have a question here from Mara about the presence of Cisco in Lake Michigan. Are there Cisco in Lake Michigan, and maybe you know a little bit about their status?
Sure. So if we, well, I won't skip all the way back to the beginning, or maybe I will. Let's see. So if we go back to this very first graph that I showed, or a picture, Cisco were in all the Great Lakes, and they still are in most of them except for Lake Erie. So Cisco themselves are still in Lake Michigan. They're just definitely not as robust of a population as they used to be.
Great, thank you. We also have a question from Ellen, who it sounds like or is a fellow researcher.
She's a fellow Cisco sister, as we like to call ourselves.
Cisco sister, right on. That's great. I really love that. um, what do you think are the next steps for Cisco restoration in the Great Lakes?
Ooh, good question, Alan. So I could probably talk a while about my personal thoughts on this, but I will give a more specific example towards that last project I discussed with the habitat. So that project was geared towards understanding where the adults are laying their eggs. And that's important to know, but the probably more important thing to know is if the eggs are actually surviving from those locations. So maybe the adults are making a choice to put them in one location, but maybe that location isn't where the eggs will actually survive. Maybe that's where the predators will eat them, or, generally the fish are well adapted to that, but with changes in habitat, their previous choices might not be the best choices anymore. So I would say the next step to that project would be to look at all the places that they're laying their eggs in the spring when they're starting to hatch and figuring out where the fish are actually surviving from. And that'll really give us a little bit more information to know what we need to do on the habitat conservation and restoration front. So that's just a particular example, but every time I do a research project, my head starts swimming with ideas on how we can use this to inform conservation and restoration. And a lot of times it's thinking about new research projects to help learn more. As you know, there's some stocking efforts going on where they're adding fish back to the lake, and those are just in beginning stages. So figuring out the success of those projects is also probably going to be an important next step as well, which is currently also being done. But learning those results will be important.
Awesome. Thanks, Hannah. There are a few more questions coming in, but we're rapidly approaching 1 o'clock. So I'm wondering if you wouldn't mind sticking around, and I can just run through the feedback poll and the last couple pieces that I have, and then maybe we can revisit folks' questions if you're willing to stick around.
Yeah, definitely willing.
Okay, great. So I'm going to go ahead and share my screen, and we're gonna jump ahead here to the feedback poll. So my colleague Caroline should be launching the feedback poll in just a moment. So if you wouldn't mind taking a second to run through this set of 3 or 4 questions and just let us know how we're doing. Give us a little bit of feedback. That would be greatly appreciated. So we'll give you all just a minute or two to run through this and give us some feedback. While you're finishing up your feedback poll, I'm just going to jump in to the take-home activity for those of you that are interested. So I think we could take a lot of this information and a lot of this learning and get a little creative with it. So maybe you'd like to tell the story of Cisco and their conservation and restoration in the Great Lakes in your own words. So write a little story, maybe make a comic, write a poem, figure out a creative way to use words and illustration to tell the story of these fish, why they're important to the ecosystem, and why they're important to people, and what people can do to help them thrive. Maybe it's we need to do more research. Maybe we want to implement some of those conservation strategies that Hannah talked about today.
Can I add a quick little plug on that? So please. I just remembered that on Twitter, there's actually a wonderful scientific community. And there's actually a lot of people who do different sketches and share them on Sunday with the hashtag, I think it's Fish Sketch Sunday or something like that. But that would be a great place to show some of your stories or sketches, I think. I think the scientific community would love to see them.
Awesome. That's great. Thank you, Hannah. So yeah, you can include that Fish Sketch Sundays hashtag. You can share your work with us on Instagram or on Facebook. We have our handles down there at the bottom. So yeah, that would be awesome. And then Up next, so this Friday, we have another Zoom of Scientists session happening again at noon on May 22nd, titled Green Stormwater Infrastructure, Incorporating Natural Systems into the Developed Landscape with Justin Geibel and Sarah Hoffmeyer. So infrastructure is the basic equipment and structures that are essential for functional, healthy, and vibrant communities. The natural infrastructure we rely on to mitigate the impacts of human development are often overwhelmed, and this is the case in managing stormwater and water pollution. Green stormwater infrastructure allows us to incorporate analogs of natural systems into our developed landscape and help mitigate the impacts of our development. Let's explore the concepts of green stormwater infrastructure technology and take a look at a real-life installation that is a small piece of a larger effort to support healthy communities and a healthy Lake Champlain. So I hope you all will join us for Friday's Zoom of Scientists session. You'll find the links to our virtual learning page to register in the chat box, as well as a link to the Lake Champlain Sea Grant YouTube page to come back to this presentation and all of our other Zoom of Scientists presentations that are archived there on the YouTube channel. So thanks again for joining us. If you have more questions, Hannah has agreed to stick around and field a few more. So Hannah, I am going to stop sharing, and you and I can just hang around and go through some more of these questions. So Emily Hunter asked, is there a native species in Lake Champlain that is being researched in this way to this extent? So perhaps using some of the similar methods maybe that you talked about. I'm not sure if you're aware of any research like that going on in Lake Champlain.
Good question. So to start, Cisco are actually in Lake Champlain, and whitefish are. They're not being researched in this way in Lake Champlain because they're just not as economically utilized as they are in the Great Lakes. But there is some interest in learning about them because they're a native fish that is important to lake trout, for example, and other species as well. But I would say in Lake Champlain, lake trout is probably a species that has a decent focus for conservation and restoration. Ellen Marsden has either talked or will be talking, and she's a wonderful lake trout scientist, and a lot of her graduate students have also become wonderful experts as well in this way. So I won't dive too much into their research just for the sake of their own presentations, but there's some really interesting stuff that's been going on in the past. And then also they're becoming a bit of a success story in Lake Champlain. So that's really great to see kind of what the mid to late stages of conservation and restoration look like, and then what the next steps are in that realm as well. So once you start to see the population come back, what decisions do you need to be making? So like I said, I won't go too much into detail just for the sake of their presentations, but it's some really interesting stuff.
Yeah, I appreciate that. And Ellen has led a session, and some of the other researchers as well here in Lake Champlain looking at lake trout and some of the other species here. So if you're interested, yeah, you can check out some of those presentations that go in depth there. Thank you, Hannah. So John has a fun question. All of your pictures show super smiling faces. Are you all that happy with your research work? And how competitive are these kind of positions for entry and to get into?
All very good questions. So I'm probably, you could tell, a bit of a smiley person in general, but I will say that I have not seen too many sad faces, except for there are some people who don't love the winter field work as much. As I said, you're out on the boat in the winter, sometimes it's snowing, you're bundled up, but it's still, some rough conditions. There's also Ellen George, who is, a participant or listening in. She has also done research where you literally walk out on the ice, and other researchers in the Rube Lab have done such scenarios as well. So I would say the field work is definitely fun regardless, though, at least for me and for a lot of the scientists. There is a lot of the lab work as well, which I described, but you didn't see as many pictures of because, it's just less exciting to share those pictures, but there's still some interesting lab work that goes on. And for me, I really like the mix of both. As for the competitiveness, I think any field has a certain level of competitiveness. I have worked previously in more lab-based research and then also more wildlife research. And I would say wildlife versus fisheries, depending on the species you're working on, it can be kind of competitive. Like a lot of people want to go study wolves, which is really cool, and you still can, but it's a little bit more competitive. Fisheries is competitive at a certain extent, but I would say they're also just a little bit more of a welcoming and supportive community. So while it's competitive and it can be hard to find long-term jobs sometimes, generally, if you connect with the right people, show that you're interested and engaged and willing and a hard worker, folks will try to help guide you through the process and guide you to the right opportunities to help you along the way.
That's great. Thanks for that, Hannah. I think I can imagine that there would be less smiles on Lake Superior when there's still like ice in the middle of July. But yeah, also I'll just add, when you enter college or even, late high school years, or maybe it's summers, there are lots of interesting professors doing great work that are always looking for a hand from undergrads, and that's a great kind of first step to start to get involved in some of these research projects and maybe get your foot in the door and start to know the community a little bit. If you're really interested in fisheries, finding a professor who's doing research and you're willing to haul nets or do whatever it is to lend a hand.
I will also add that if you don't know if it's something you're interested in, try it anyways. Like that's how I kind of figured out what I liked, was trying a bunch of different things. And I didn't have as many opportunities as a high school student to do these things. So a lot of my investigating was in college, but I agree, high school is a great place to start. And one other avenue I'll mention is citizen science. So there's a lot of great projects where maybe you're not working directly with a researcher out on the boats, but if you go fishing yourself, sometimes they need to know what fish you catch and where and when and the size. So just kind of like do a little search and see if there's any way that you can contribute your daily activities towards science, and that connects you with researchers, and therefore it also might put you on their radar for when opportunities do come up to help with more in-depth research. So that's another good way to go about it.
That's great. Yeah, thank you for that. We have one more question from Mara again. You said there were about 4 species of Cisco. I think you highlighted a few others as well. Do you think there are more species of Cisco? So perhaps some undescribed species. I know you had some of those hybrids that were unnamed.
Yeah, so that's a good question. Historically, there were definitely more than just 4. Unfortunately, some populations took a harder hit with those declines that I described early on, and some of them are completely extant, which means they're gone completely now from the Great Lakes. So sadly, that's true. Perhaps, you always hear stories of, this rare bird was rediscovered in the Galapagos when we thought it was extinct. So, who knows? It's a huge lake. Maybe they're just dwelling in some parts of it we can't get to, but the likelihood is that, we might have come across it by now. So maybe some of those older forms are still there. It is possible that over time they might develop into new forms or species, because this fish, as you can see, close relatives have adapted to be in a wide range of roles in the ecosystem. So part of that is, if we talk about genetics briefly, they have 4 copies, at least 4 copies. They have multiple copies of their whole genome. And that gives more material for adaptations to occur. And that's a whole nother topic, but basically they're well suited to adapt, and hopefully that'll help them adapt to the future. And it could mean adapting, one fish adapts this way, one fish adapts that way, and then they become different species. So it is possible that in the future there will be additional species, but yeah, I don't know about currently. It's hard to know.
Yeah, thanks for that. Continued research, I suppose. Continued monitoring. Great. That's all we have for questions. I'm gonna drop your email and your Twitter handle into the chat box. So if folks do have subsequent questions, they can reach out to you directly. And I just want to thank you for giving a lovely presentation and sticking around for some extra time to answer some of those questions. And thank all of the participants for showing up today. So thanks again, Hannah. Really appreciate
Yeah,
it.
no, thank you all for having me. It was fun.